Okay, this video is going to go over Chapter 25, Objective 3, Part B, or Part 2. So we're going to be looking at the second process of urine formation called tubular reabsorption. So this one's going to be probably about 15-minute video. So again, we're going to be looking at our picture here. Tubular reabsorption is going to be occurring in this Part 2 area right here. So remember, reabsorption is going to be where materials, once they've been filtered through the process of filtration number 1, next we're going to return some of those things back to the bloodstream. So this is tubular reabsorption. So tubular reabsorption is the return of filtered water and solutes back to the blood. So we've got our capillaries right here, and you can see that this is going to start immediately after filtration has occurred, and this is going to carry on through the rest of the nephron. So we're going to be taking a look at tubular reabsorption throughout the entire nephron and what is being filtered or what is being resorbed where. So there is a review if you need to take a look at simple, facilitated, and active transport again. This is the movement of materials, simple and facilitated, from an area of high concentration to an area of low concentration, just by itself or with the help of a protein, but it's moving things from high to low. No ATP is needed. Then you have active transport. Active transport is the reverse. You're moving things from an area of low concentration to an area of high concentration, and that's going to require some ATP to do that. So in order to move things from the nephron into the blood, return it back to the blood, it's going to rely on these three processes: simple, facilitated, and active transport. So to review that, if you've forgotten, Khan Academy has an awesome review on those three processes. So tubular reabsorption is the return of that filtered water, the water that came through the filtrate, and solutes, and returning it back to the blood. Methods by which this occurs: again, simple diffusion, facilitated diffusion, and active transport. And these are hyperlinked within the PowerPoint, so I will let you take a look at that on your own if you need to review those. When we're talking about active transport, the primary active transport method that we're going to be looking at is the sodium-potassium pump. Again, this should be review for everybody. We've talked multiple times about the sodium-potassium pump. The key again is we are using ATP to move things because you're moving them against their concentration gradient. There is some secondary active transport mechanisms that are being used. Symport. So you've got a little visual here. Notice both particles are moving together. You've got two particles moving from one side to the other side. They're moving together. One ion is going with its concentration gradient, meaning you're going to need some energy, while the other is moving in the same direction against its gradient. So one of these particles is needing energy to move because it's moving from low to high. The other one is moving from high to low and doesn't require energy. So symport, you've got, while they're both moving in the same direction, one is moving with the gradient, the other's moving against. Then you have antiport, which is primarily a sodium-calcium exchange mechanism. This one, antiport, they're moving in opposite directions. Now, you're still moving one along its gradient and one against its gradient. But the only thing is they are moving in opposite directions. So understand the definitions of symport and antiport. And then also osmosis, which again is the movement of water from an area of high water concentration to an area of low water concentration without any energy expenditure. So this is a very busy handout right here, but let's just kind of take a look at some things. Let's kind of break it down. This video, the Khan Academy video, does go over this specific picture right here. There is a handout in your packet. It's about an 18-minute video for Khan Academy. So let's just kind of look and see what's happening here. First off, you do need to understand the different parts of the nephron. So you should be learning that in lab, but just to quickly go over that, you've got your afferent arteriole going in, efferent arteriole coming out. So this capillary within is that glomerulus. It resides in Bowman's capsule, and all of it is the renal corpuscle. Now, you've got your black arrow right here, which is representing the filtrate going into this first section of your nephron. Now, this is stretched out, okay? The nephron actually doesn't look like this. It's all kind of intertwined, but again, just to kind of show you where all the parts of the nephron are, this nephron has been kind of stretched out a little bit. So you've got this area right here. Notice it's kind of convoluted. It's kind of wiggly. This is your proximal convoluted tubule. Now, to understand why it's called proximal, remember proximal is going to be, when we used it in the appendicular skeleton, it was closer to the point of attachment. So what are we talking about here? So we're talking about closer to the glomerulus. Okay, so it's proximal to that. So this is your proximal convoluted tubule. Then you're going to go down the descending limb of the loop of Henle and up the ascending limb of the loop of Henle. You can see the black arrow showing you direction of movement. And then you're going to go into the distal convoluted tubule. Now, in reality, the distal convoluted tubule is going to be way over here next to the glomerulus, but as the fluid travels, right, it's further away. And then from the distal convoluted tubule, the material at this point is pretty much called urine, and it will enter into the collecting duct. So you have all these little stems right here, which are distal convoluted tubules of other nephrons. So you have a bunch of nephrons dumping their urine at this point into that single collection duct. Okay, so now that we kind of have an idea what's going on, this purple structure is your kidney. And if you remember, your kidney is divided into two main layers. There's that little bit of connective tissue that's on the very, very outside. We're not going to be talking about that. But this area right here, which is on the outermost layer of the kidney, this is going to be your renal cortex. And then this area down here is going to be the renal medulla. So you can see the nephron spans both the cortex and the medulla. It's in all parts. You can see right here kind of where we're looking at there. Okay, so now let's kind of get into what's going on in each of these parts. Looking at the proximal convoluted tubule, you can take a look at these arrows to see the direction things are moving. I am not going to ask you on the test under what process it's moving, so you don't have to necessarily know the colors of the arrows. The big thing is I'm going to be asking you what's moving in what direction, okay? So you are going to lose, that should be an L right there. That looks like an I, but that should be an L, sodium chloride. So sodium chloride is going to be returning to the blood. Bicarbonate ions are going to be returning to the blood. Water is going to be returning to the blood. And then here's the biggie. Glucose and amino acids, the building blocks of carbohydrates and proteins, are going to be returning to the blood. So keep in mind, all of that got filtered out based on particle size, but we want to keep all this stuff. We want to put it back in the bloodstream. You also are going to have some drugs and toxins that are going to enter the proximal convoluted tubule and some hydrogen ions that are going to enter the proximal convoluted tubule. So you're going to get a lot of reabsorption at the proximal convoluted tubule. A lot of it's going to get reabsorbed. Now, from there, you're going to go down the descending limb of the loop of Henle. Take a look at what is being reabsorbed at the descending limb. Water. I need you to understand that. Water is what's leaving the descending limb, and then look at what's leaving the ascending limb. Salt, sodium chloride, okay? So again, I'm not going to ask you processes on how it's moving. I just need you to understand what's moving. So descending limb, water is going to leave the nephron and enter into the blood, and then sodium chloride is going to leave the nephron and enter into the blood on the ascending limb. And then from there, we get into the distal convoluted tubule, and you're going to have some more sodium that's leaving, some more bicarbonate that's leaving, and then you're still going to have some drugs and toxins entering in and some hydrogen ions entering in. Now, when you get over here to the loop of Henle, the collecting duct, take a look at what's leaving. Water is leaving. Understand that what is controlling water leaving the collecting duct is going to be your antidiuretic hormone, also known as vasopressin, right? So antidiuretic hormone, if it is present, it's going to open these channels right here and allow water to exit, which means that's going to decrease urine volume, right? If you don't have any antidiuretic hormone present, The water will stay in the collecting duct and will leave the body in the way of urine. So antidiuretic hormone present, less urine will be made because you're going to resorb that water. You're also going to get some sodium chloride leaving and some urea that is leaving as well. So this just goes around and talks about each piece of this individually. The big takeaway that you're going to need to know is in the proximal convoluted tubule, what is leaving. So you've got salt, bicarbonate, water, glucose, and amino acids. The descending limb, extremely permeable to water. So you've got water exiting the loop of Henle, going into the bloodstream. And again, anytime you got water, it's pretty much osmosis. In the ascending limb, you're going to be losing sodium chloride from the kidney back into the blood. This is active transport, so that's going to require some ATP. But again, for the most part, just know sodium chloride is leaving through the ascending limb. And then you've also got as you move up, you still have sodium chloride that is leaving. It's just leaving a different method, but we're not going to worry about that for the test. Distal convoluted tubule, you're going to have sodium chloride leaving, you're going to have bicarbonate leaving, and you're going to get some drugs and toxins and hydrogen coming in to the nephron. And then the collecting duct, again, you're going to have some sodium chloride involvement here with Symport acting. Water is resorbed when antidiuretic hormone is present through the process known as osmosis, and that will result in a very concentrated urine. This is an awesome video, again, for you to watch. This is an Armando Hasudigan video, and he goes through and explains just what we talked about. So if you need a visual, he does a really great job of explaining the nephron and the resorption part of that. Again, Khan Academy does a great job as well. Okay, so the last part is tubular secretion. So we'll do that in the next video.